Bismillahirrahmanirrahim. Dear students and audience, in this video we will talk about at introductory level on the subject of animal genetic resource conservation, which is a growing issue in the area of biology and more specifically animal biology. Now, what are animal genetic resources? They include all breeds, types, varieties and populations of animals that are living on the face of earth. It could be in improved or unimproved conditions. The reference material for this stuff is available in two FAO documents. FAO 2013, it talks about in vivo conservation, and FAO 2012, it is cryoconservation of animal genetic resource. Now, first thing is, how can we define a breed? A breed are animals that through selection and breeding have come to resemble one another and pass those traits uniformly to their offsprings. More simpler definition is, a breed is a group of domestic animals termed such by common consent of breeders. And another breed definition is, breed is a breed if enough people say it is. So breed defining is prerogative of breeders. They will have to define the breed. For more advanced breeds, breed associations are there who define a breed precisely. Now, what are breed standards? These are a description of characteristics of ideal animals to be obtained through breeding program of a standardized breed. So we would be looking for an ideal animal within a breed, and on that ideal animal we will set the standards for that breed. This is typically a job of a breed association or breeders who will write breed standards with the help of academia or animal scientists or animal breeding experts. And a breed is considered at risk once its population becomes very low in number. For example, we start thinking that it will fail to exist in future unless we start a conservation program to save that breed. So that breed is considered at risk. I will talk about the various classes of risk in a different lecture, but for now we just need to assume that if breed has very limited number of population size, we can think it is a breed at risk. And census size is simply population size. It is the number of living animals in a population. It is slightly different from the effective population size, which is a number of breeding males and females of a breed, and it accounts for genetic relationships among animals. One more question is, who decides or who registers a breed at international level? There is no international body for breed registration. It may be registered at country level. For example, in Pakistan we have Animal Husbandry Commissioner, which is one of the 5 technical wings of Ministry of National Food Security and Research, Government of Pakistan. These guys are responsible for registering a breed. Similarly, in case of India, they have a National Bureau of Animal Genetic Resources. They have notified a breed registration committee. Anyone can apply for registering a breed. They register a breed on the basis of 2 criteria. Population must have been bred pure for 10 generations, and scientific evidence of their uniqueness and reproducibility. So a breed has to be registered at a national level. There is no international body for that. Now, coming back to basic definition of heterozygosity, it is a condition where 2 alleles at a given locus are different. Heterozygosity is generally advantageous because a favorable allele can often compensate for the effect of an inferior or detrimental allele at the same locus. and usually mean heterozygosity is used as a measure of genetic diversity. And as opposed to heterozygosity is a condition in which both alleles at a given locus are same. They have received same thing from both parents, so the individual becomes homozygous. And homozygosity is generally unfavorable. It is a threat to genetic diversity. Now, it is easy to define genetic diversity. It is a range of genetic differences among organisms, and we have two criteria of measurement of genetic diversity in livestock. One is number of breeds within species. If we have more breeds within a species, we have increased genetic diversity. And similarly, we need to look at level of heterozygosity within breeds. So animals within a breed should have genetic differences, and similarly there should be more breeds within a species if we need to set a genetically diverse population. Now, what are the factors that threaten animal genetic diversity in the country? It is a little bit different in case of developed world. Developed world have a growing trend or reliance on very limited number of international transboundary breeds, which are suited to high input, high output animal industrial agriculture. The example is Holstein dairy cattle, which is international breed found everywhere in the world. But the thing is, when we have more animals of one breed, then the use of the other breeds becomes at risk, and this trend has a negative impact on animal genetic diversity. In case of developing world, we have a whole lot of issues similar to human populations. For example, climate change, indiscriminate crossbreeding is going on, trends in consumer demand, and we lack infrastructure and services for breed improvement. We do not have labor force and traditional knowledge, and there are a range of disasters which threaten the existence of a breed. So what are the objectives of conservation? They are economic. It will allow for greater response to selection and faster adaptation to changes in climate and production systems. Animals are affiliated, humans are affiliated with specific breeds, and there are clear social and cultural objective of conservation. In case of environment, we need more adopted breeds, so that is one of the objective of conservation. Similarly, risk reduction, any risk to food security can be reduced by having more and more diverse animal populations. And similarly, for research and training in the fields of nutrition, reproduction, immunology, genetics, and adaptation, we need to maintain and conserve genetic diversity among animals. We have a couple of methods for conserving animals. Conservation of live animal populations, for example, which is called in vivo conservation. In this conservation, we have two types: in situ and ex situ. In situ is on site or in the original place or in the production system, and ex situ is off site or away from production system. So in situ conservation is keeping the breed in their original production system where they were evolved. Here we have a challenge of making them more productive and competitive. And in ex situ conservation, we keep animals away from their production system, for example, zoological parks and government farms. So it is hard to define between these two distinctions. Similarly, cryoconservation is not keeping the live animals. Instead, we keep their genetic material, for example, semen, embryos, and somatic cell, so that we can reconstitute those animals again if we need them. So this is the basic introduction of animal genetic resources and how can we conserve them. I will talk about more specific stuff in the later videos and how we can conserve it. Thank you for your attention, and keep watching these informational stuff.